for the structure of peptides. Good place to start, right? All right, let's get started with the structure of peptides. And if you remember in our last class, we talked about amino acids and how an amino acid obviously contains an amino group and an acid group. And we talked a little bit about amide bonds. And I said that when you have an amide bond between two amino acids, we call it a peptide bond. And so we have an example of a peptide down here. This is called glutathione. Sometimes it's abbreviated as GSH, and it's an example of a tripeptide. Why? Because it's made from 3 amino acids. It's made from a glutamic acid, a cysteine, and a glycine, and they're all linked together. And what are the linkages that hold them together? An amide bond and an amide bond, right? 2 amide bonds, but we call those peptide bonds because they link these 3 amino acids together. Now, we have the glutamine, we have the cysteine, we have the glycine. These are the 3-letter abbreviations that are shown right here. You can also use the 1-letter abbreviations. Again, I don't expect you to memorize those. For this tripeptide, it would be E for glutamic acid. Then we have C for the cysteine, and then we have G for the glycine. So again, E, C, G. In this class, you're not expected to memorize them, but there they are. Now, what I want to point out is that it isn't enough to just join these 3 amino acids, glutamic acid, cysteine, and glycine. They have to be joined in the correct order in order to make GSH, in order to make glutathione. For example, let's say we had them organized, like let's say we had the cysteine, and then we had the glycine, and then we had the glutamic acid. That is not GSH. It isn't. It's something else. It's a constitutional isomer of it, but it is not it. Again, let's say we had glycine, then cysteine, and then glutamic acid. Glutamic acid. Again, that is not glutathione. That's something else. These would be constitutional isomers. Constitutional isomers of glutathione. If you're wondering why did they pick this tripeptide to put in the textbook, glutathione is a pretty interesting compound. It's a scavenger of harmful radicals in the body and harmful electrophiles as well. And the reason why is because it will form what's called a GSH adduct or a glutathione adduct because it has a thiol in it, and thiols are really good nucleophiles. But that's probably something that you'll cover in your biochemistry class. Well, let's move on from there and talk about a dipeptide. So let's say we want to make a dipeptide. Let's say we want to make a dipeptide from alanine and glycine. So remember the R group in alanine, its R group is a methyl. And the R group in glycine, well, there is no R group really, it's just 2 hydrogens. So there we go. What are the possible ways you could put a molecule of alanine and glycine together? Well, you can probably see on the slide that there's 2 ways you could put them together. You could have an amide bond or a peptide bond formed between the carboxyl group of alanine and the amino group of glycine. That would give you this compound. So alanine on the left, glycine on the right. Remember, we always have the nitrogen on the left-hand side. Or you could have them bound with the glycine forming a peptide bond, the glycine's carboxyl group rather, forming a peptide bond with the amine of the alanine. And again, you can see that these 2 compounds, whether you have alanine attached to glycine or glycine attached to alanine, these are 2 constitutional isomers, okay? These are constitutional isomers. They are not identical, all right? So you notice that I said that the nitrogen was on the left-hand side. Well, that's actually a convention that we use in organic chemistry. We always have the nitrogen on the left-hand side and the carbon on the right, we always have the nitrogen on the left-hand side and the carbon on the right-hand side. This is a convention. Just like reading the English language, we go from left to right, and the nitrogen on the end, we call it the N-terminus. Sometimes we call it the N-terminal. And then on the right-hand side, we have the carboxyl group, which contains the carbon, and we call that the C-terminus. But the bottom line is the N-terminus is always written on the left-hand side, and we're going to get some practice on that today. Even if we use those three-letter abbreviations, like you see here, we have alanine, glycine, cysteine, tyrosine, glutamine, phenylalanine, cysteine, again, methionine, the name escapes me at the moment. And then we have leucine. So again, the N-terminus is going to be on the left-hand side, and the C-terminus is going to be on the right-hand side. So a little bit more about this whole peptide bond thing. This goes all the way back to, chapter 1 and chapter 2 of Organic Chemistry 1. We discussed this very concept right here back then when we were, probably would have been in chapter 2 because we were discussing resonance back then. But it says here, recall that peptide bonds are amide bonds. I think we have that down. A peptide bond is an amide bond. But the carbon-nitrogen bond in an amide, so this bond right here, has partial double bond character. You can see that when you draw all the contributors to the resonance hybrid. Look at this one here. We have a double bond between the carbon and the nitrogen. And also in this one, everything has a complete octet. So this is definitely a contributor to the resonance hybrid. And what that means, again, is that that carbon-nitrogen bond in our amide has partial double bond character. And because of that, there's a high barrier of rotation for the carbon-nitrogen bond. For this bond to rotate, it takes a high activation energy of 80 kilojoules per mole. And what the consequence of this is that when you make peptide bonds, a whole bunch of them, or even just one, between two amino acids, what happens is that the two groups that are attached to the nitrogen and the carbonyl, they want to get as far away from each other as possible. And we call that the S-trans conformation. When it says S-trans, what that means is sigma. So sigma trans, because this is a sigma bond. It's not like a double bond cis and trans. It's sigma trans. You can even scribble that in here. Sigma trans, and then you have sigma cis. Okay, the same thing over here, S cis and S trans. But give me a thumbs up if you follow me on the idea that the two R groups here, so to speak, are farther away from each other, and thus this would be more stable. The S-trans conformation. It's actually a rotamer is what it's called, but we'll just put here more stable. All right. Well, let's move on from there. And if you look at a peptide and you look at the backbone of a peptide, yeah, if you look at the backbone of a peptide, so you see that you have 3 different peptide bonds here. Well, those are the only bonds in the backbone that experience that limited rotation, that restricted rotation, because they have partial double bond character. All of the other bonds, this one, those experience free rotation, no problem whatsoever. And because of that, it enables a peptide to adopt a variety of conformations. And this is why peptides fold in on themselves and form some really interesting structures. And you might have seen those when in biology, if you've studied the tertiary structure of proteins. Something else that's interesting that peptides can do is they can form what are called disulfide bridges. So hopefully you remember from way back in the day when we talked about functional groups that if you have SH, that's called a thiol. So maybe I'll write it down here. This is a thiol, and this is another thiol. And thiols are found on cysteine. So this is a molecule of cysteine, and this is a molecule of cysteine like this. And what happens is they can undergo an oxidation process. How do we know this is oxidation? Because you're losing hydrogens, right? You lose this hydrogen and this hydrogen, and you form a bond between the two sulfurs, and we call that a disulfide bridge. A disulfide is when you have an R group with 2 sulfurs and then another R group. This molecule, so this one is called cysteine, okay? This one is called cysteine. This molecule is called cysteine. But that's a disulfide bridge, and we take advantage of disulfide bridges in hair when we make a perm. So if you've ever heard of anybody having a perm before, well, what they're doing essentially is first they're reducing the disulfide bonds in their hair so that all the disulfide bonds are broken. Then they wrap it around a curler, okay, like this. So there's the curler, and then they oxidize it so that they form out the disulfide bridges, and then they just take the curler out and the hair remains curly. Anyhow, I don't know what that's got to do with. I probably won't be quizzing you on getting your hair permed, but now you know how it works. Anyhow, another thing about these disulfide bridges is that they can greatly affect the three-dimensional structure of a protein. You link all these amino acids together, but you can imagine that if you have two cysteine residues linked together by a disulfide bridge, that will create this kind of turn like this in the molecule, and that greatly limits the freedom of movement of the chain, so to speak. We can actually have a disulfide bridge between two cysteine residues within the same chain. That's called an intra-strand disulfide bridge. And if they're between two cysteine residues and two neighboring molecules or two different strands, we call that an interstrand. So inter-strand disulfide bridge and intra-strand disulfide bridge. Make sure you know the difference between the two. We have an intra-strand disulfide bridge. This isn't the structure of insulin, but you can find one in insulin if you look up the structure of insulin. All right, the whole thing about biologically relevant peptides, I'd say this is more of an FYI than anything. And I think the main point here is that you understand that very small changes to the sequence of amino acids can have drastic effects on the activity of a compound or the target of a compound. So these two compounds are called enkephalins. You have leucine enkephalin, which has a tyrosine, a glycine, a phenylalanine, and a leucine. So leucine enkephalin ends with a leucine, right? This is the N terminus, this is the C terminus. And then if you replace the leucine with a methionine, then you have methionine enkephalin, and both of these function in the brain in the control of pain. They're both believed to bind to the same receptor as morphine, and we would classify these as pentapeptides. So we'll put in here pentapeptides because they both possess 5 amino acid residues in their structure. The next one that we have here is bradykinin, which acts as an anti-inflammatory agent by dilating blood vessels. And you can see that it's got all these amino acids linked together. So we have an arginine, proline, glycine, phenylalanine, serine, proline, phenylalanine, arginine. You do not have to have the structures of these memorized whatsoever. It's not part of the course. It's, again, more of an FYI thing. So bradykinin. Next, vasopressin and oxytocin. If you look at these two molecules, so here's vasopressin, this is oxytocin. They both possess an intra-strand disulfide bridge. They both have most of their amino acids in common. You can see that we have a cysteine, tyrosine, but you see that they have a difference in the next residue. Here we have a phenylalanine, here we have an isoleucine, and the only other difference is this arginine and this leucine here. But they both have very different biological effects. For example, vasopressin deals with or has something to do with blood pressure, and oxytocin induces labor and stimulates lactation after birth. Another thing is that if this is our N terminus, the N terminus usually ends with NH2 unless it's got a proline on the end. But the C terminus usually, usually ends with a carboxyl group, doesn't it? But here, in this slide, they're showing that it's got an NH2. So what that means is that the C terminus, in this case, is actually, we'll just write it like this. It's a carbonyl, but then it's got and NH2. So it's a primary amide on both of these, okay? Anyhow, that's just what they're showing here with these NH2 groups on the end. It's not an OH that ends in the molecule. It's an NH2. Well, that's enough about the structure of peptides. Let me show you something really neat. Let me show you how to draw the structure of a peptide. So it says, draw this tripeptide. So it's leucine bound to alanine and then glycine. Well, first thing we have to remember is this. Follow me very carefully here. You always start with the N terminus on the left, and then you go to the C terminus on the right. So since all the amino acids that we're looking at here are alpha amino acids, right, where we have an R group, and then we have an NH2 like this, we're always going to go nitrogen, carbon, carbon. Okay, so NCC, NCC. Now, if you want to be even more detailed, you can say NC, and the next carbon is a carbonyl. So since we have 3 amino acids, watch very carefully. We're going to go nitrogen, carbon, carbonyl. Nitrogen, carbon, carbonyl. Nitrogen, carbon, carbonyl. Okay, those are our 3 amino acids, right? Each amino acid always goes nitrogen, carbon, carbonyl. Okay, they always have that order. So on the N terminus, we're going to put our NH2. On our C terminus, we're going to put our OH. Now we're going to put in our R groups. Here's the key. When you're putting in the R groups, so for leucine, the R group is an isobutyl. If the R group is pointing down, in order to get the correct stereochemistry, which is S, you want to put anything that's going down on a dash. So there's our isobutyl. Then we go to the R group of alanine. Well, anything that's going up, we're going to write that as a wedge. Everything. Okay. Any amino acid side chain that's pointing up, we write as a wedge. And the next one is a glycine, and glycine doesn't have any R groups. It's just 2 hydrogens. So there we go. The next thing that we have to do is fill in any missing atoms. You can see that we're missing an atom on this nitrogen and this nitrogen, and there you go. We've drawn the structure of the compound. There's nothing wrong with what I've drawn here, but if you want to make this Zwitterion and say, hey, this is a physiological pH, so you'd put NH3 plus here and O minus, then that's fine too. I had a student ask me a couple of semesters ago, and I thought it was a good question. She said, okay, well, if it's, if this any R group pointing down is on a dash, what would happen if you had started, kind of in a different way? So what she meant was this. What if I had gone nitrogen, carbon, carbonyl, nitrogen, carbon, carbonyl, and then nitrogen, carbon, carbonyl, like that? What if I had done that? Would the same thing still hold true? Yes. Now your R group here, your isobutyl, would be on a wedge, and your R group here would be on a dash, like that. But both of these are identical. Okay, there's nothing wrong with either of these. They're both the exact same molecule. So remember, R group goes up. We'll say R group pointing up is a wedge. And if it's pointing down, it's on a dash. And that's it. All right. Give me a thumbs up if you follow me on that. If you're thinking, Mr. Dean, I can draw a peptide. I could do that. Everybody with me on that one? Drawing a peptide? Cha? No? Okay. All right. Let's take a look at another problem then. It says, using 3 and 1 letter abbreviations, show the sequence of amino acid residues in the following pentapeptide. Remember that if it's a pentapeptide, it's going to have 5 NCCs, right? We're going to go nitrogen, carbon, carbonyl, nitrogen, carbon, carbonyl, et cetera, et cetera. So here you see nitrogen, carbon, carbonyl. That's your first amino acid, this one right here. Then we have nitrogen, carbon, carbonyl. There's your second amino acid. Then we have nitrogen, carbon, carbonyl. That's our next amino acid. We do it again. Nitrogen, carbon, carbonyl. And the last one, we have nitrogen, carbon, carbonyl. Again, if you're wondering where I'm getting those, it's nitrogen, carbon, carbonyl, I'm going along the backbone right here. Notice that everything that's pointing up is on a wedge. Everything that's pointing down is on a dash. So that's all good. But now we're going to try to identify these amino acids. Could anybody tell me the name of the amino acid when I have an isobutyl group as my R group? It's not a trick question. You could look it up in the handout, the amino acid handout. Anybody know the name of that one? No? Okay. So that would be a leucine. So we'll write leucine, which is LEU. If we look up the one-letter abbreviation for leucine, it's an L. The next one, we have a methyl, just in a methyl group. So that's an alanine, and the one-letter abbreviation is A. The next one is a phenylalanine. Phenylalanine has PHE as its three-letter abbreviation, and the letter F for its one-letter abbreviation. The next one is cysteine, which is CYS, its three-letter abbreviation, and capital C for its one-letter abbreviation. And the last one is aspartic acid. And aspartic acid is ASP, and its one-letter abbreviation is D. So if you're wondering, I always used to get asparagine and aspartic acid mixed up. Asparagine is N, and aspartic acid is D. So there you go. We've put them all together. So again, we have leucine, alanine, phenylalanine, cysteine, and aspartic acid. So again, you don't have to memorize any of the amino acids in this class. I would recommend trying to memorize a few, especially if you're going to take biochemistry. It just makes studying a little bit simpler. But there you go. Give me a thumbs up if you think you could identify some amino acids. If I said, here's an octapeptide, could you identify a few amino acids? Anybody? All right. Good. Thank you. Okay. Okay, thanks, Caitlin. I'm glad. I think you could too. I know you could. I know all of you could. I just want to be double sure that nobody has any questions. And again, if you do, just stop me. I'm happy to talk about it. Well, let's go back here. Let's say you had this pentapeptide. Okay, somebody gave it to you. Let's just imagine you're in the organic lab. It's a white solid, and somebody says, "I know this is a pentapeptide, but I don't know what amino acids are in it. Can you figure it out for me?" You can imagine that if you ran an NMR on this thing, you would not be able to figure out very much at all because there's so many protons and so many of them have similar chemical shifts. I mean, so many things would be overlapping. It would look like quite something. So how do we figure out what amino acids are in a peptide, a small peptide like that one? Well, the way that we determine the amino acids that are in a peptide, we call that sequencing. So sequencing a peptide, finding the identities of all the individual amino acids. And one of the ways that you can elucidate the structures of the amino acids in a small peptide is to use what's called the Edmund degradation. And the Edmund degradation works by plucking off each individual amino acid, like I'll pull you off, then I pull the next one, and you convert them into something called a phenylthiohydantoin derivative or a PTH derivative. And then you analyze that by NMR, proton and carbon and mass spec. And that is something that you can do pretty quickly because it's not nearly as complex as this giant peptide, for example. So here's how it works. What you do is you're going to take your peptide, and this is written in reverse, right? Because normally we always wrote our peptides where we had the N terminus on the left-hand side, and then we had the C terminus on the right-hand side. Well, what they're showing is actually the N terminus. Okay, that's the N terminus there. So they kind of flipped it around. But you know that nitrogen is a good nucleophile, and it will react with this molecule here. This is called phenyl isothiocyanate. Wrap your head around that one. So phenyl isothiocyanate, and after it reacts with the phenyl isothiocyanate, it's treated with trifluoroacetic acid. So this is, maybe I should write these names down. This is trifluoroacetic acid, and this is phenyl isothiocyanate. Okay, so you end up ripping off everything that's in the green square here. You end up plucking off that amino acid, but the amino acid with its R group, here's the R group, it gets incorporated into this molecule called the phenyl thiohydantoin derivative. And when you run the mass spec on that, or the NMR, you'll be able to figure out, hey, this is the amino acid that got ripped off. And then you just keep doing it over and over and over and over and over, because now you've got a new N terminus exposed every time you pull one off. This thing has even been automated. There's even sequencers that just do this for you. However, it's only good up to about 50 amino acids attached together. After that, it's too cumbersome, okay? And again, you're talking to somebody who's not an expert in amino acid sequencing, but there are limitations to the Edmund degradation. I am not going to ask you the mechanism of the phenyl thiohydantoin derivative formation. However, I do have it in the slides. Here it is right here. And again, I'm not going to go over the whole thing with you. It's a pretty lengthy mechanism. It's something I would never ask you. I've never seen it on an ACS. It's not going to be on an MCAT or anything like that. But if you want to, try to figure it out on your own, be my guest. But I figure at this point in the class, we've all learned enough. So what you need to know is that when you treat your peptide with phenyl isothiocyanate followed by TFA, you end up removing one amino acid. It gets incorporated into a PTH derivative, and that's it. Then you can analyze that derivative to determine its structure. Yeah, Nicole, it is a relief, isn't it? That's a long one. Look, Nicole, I'll do you one better. Look. Will not ask. Okay? You heard it. Now you've got it in writing. Okay? So you can, if I ask that, you'll be like, hey, wait a second. I won't. Okay? All right. Now you're probably wondering, well, what do you do if you have more than 50 amino acids all strung together? What if you have 150? How do we figure out the structure of some of these giant proteins and enzymes? Well, we use the process of enzymatic cleavage to elucidate the structures of proteins. If you have more than 50 amino acids, what you've got to do is break them down into manageable chunks or chains using a biological catalyst called a peptidase. And you can imagine, especially if you've studied biology, A's is something that acts as an enzyme, and peptidase is probably something that cleaves a peptide bond. And if you thought that, you would be exactly correct. You see that right here. It says a given peptidase is selective for specific peptide bonds. So it looks at all the peptide bonds and it says, I'll cut you, and I'll cut you, and I'll cut you, but only specific ones. There's two enzymes that we are concerned with in this class, trypsin and chymotrypsin. You might have heard of them if you studied proteases and peptidases in biology class. It could be, but now we're going to get into the nitty gritty of exactly which bonds they break. So here's what you need to know. Trypsin is the first one. It's an enzyme that will cleave a peptide bond, but it cleaves them on the C terminus end of two amino acids, arginine and lysine. Well, what do those two amino acids have in common? You probably noticed that they're both basic, right? Both are basic amino acids. We could even scribble that in here. Basic, basic side chains. Yeah, they both have basic side chains. Let's say you have this big old peptide right here. Okay, what you're going to do is you're going to put it in a flask, and then you mix it with trypsin. And trypsin is going to work its magic. And remember that this side of the lysine is the N terminus, And this side of the lysine over here, that's the C terminus. What did it say? It's going to cleave on the C terminus side of arginine and lysine. So it's going to break the bond in the two places that you see here where the red squiggle marks are. First between the lysine and the proline, and then between the arginine and the serine. Then what you could do is you could just do an Edmund degradation on these and kind of break them off, piece by piece, and kind of figure out what their structures are. You could do it that way. That's one way. But there's an even better way, I think, of determining the structure of a peptide this size. Check this out. Chymotrypsin is another enzyme. It catalyzes the hydrolysis of the carboxyl end, just like trypsin did, but of aromatic amino acids, phenylalanine, tyrosine, and tryptophan. So again, tyrosine, maybe I have this on another slide, I don't remember. Tyrosine does arginine and lysine, so arginine and lysine. And then we have chymotrypsin, I'll just put here chymotrypsin. It does phenylalanine, tyrosine, and tryptophan. All right, there we go. So look at this. Now you're going to take that same peptide that was on the last slide, the same one here, and you're going to do the same thing. You put it in a flask with trypsin, and you get all of these shorter peptides. Look, it breaks on the C terminus side of phenylalanine, so it's going to break here. Tyrosine, going to break here. Tryptophan, so it's going to break here. Everybody with me on that so far? All right. So now what we're going to do is we're either going to sequence these using Edmund degradation, or we could compare these fragments to these fragments because we treated the same peptide once with trypsin, then with chymotrypsin, and we can kind of sleuth our way through it to elucidate the structure of the peptide. Let me show you how you do it. Let's take a look at this example. It says you've got a peptide that's got 22 amino acid residues and is treated with trypsin to give 4 fragments. So these are the trypsin fragments right here. Then it's treated with chymotrypsin to yield 6 fragments. Identify the sequence of the 22 amino acid residues in the starting peptide. Well, remember that trypsin, let me see here, I'll just put them up here. Trypsin does arginine, put it in blue, does arginine and lysine, whereas chymotrypsin cleaves on the C terminus side of tryptophan, tyrosine, and phenylalanine. Now let's look carefully. Let's start with the trypsin fragments. On the C terminus side of arginine and lysine, we have an arginine on the end here, a lysine, an arginine, and a valine. Well, the valine couldn't have been cleaved. And we know that the valine's N terminus is here, and its C terminus is there. Therefore, the valine must be the C terminus, right? This must be the last amino acid in the chain of the 22 amino acid peptide. Look, if we go over here, we see the same thing. We have tryptophan, tyrosine, phenylalanine, we have a valine again. Oh, it's that same valine that we saw before, because we know that it did not cleave on the C terminus side. Therefore, this must be the last amino acid. And we notice that here you have an alanine that precedes it, and we have the same alanine over here. So now we're going to go backwards using all these fragments, and we're going to figure out the structure of the whole thing. So we're going to start on the right-hand side of the molecule with the C terminus. Let's begin. We're going to start with valine and alanine. So we can cross these off. We can cross these off because this is the only place that we see, no, it's not the only place that we see valine and alanine, is it? Yeah, but it's the only place that you have the valine on the end. So this must be the C terminus. So then we're going to go, after the valine and alanine, we're going to go the phenylalanine, proline, and glycine. So we're going to go phenylalanine, proline, and glycine. So we can cross these three off. Do you see glycine and proline anywhere over here? The only place that we see them are right here. We see phenylalanine, proline, and glycine. So before that, it must be arginine, cysteine, methionine. So we can cross these off. Okay. Where do you see a methionine with a cysteine and an arginine? You see the arginine, cysteine, and then the methionine here. So we pick it back up here again. So we have the methionine. After that, we have phenylalanine, histidine, tryptophan. So phenylalanine, histidine, tryptophan. Go. So we can cross all these off. The phenylalanine histidine must be this right here, because that's the only place we had it. Then this is the only tryptophan in the whole thing. So before the tryptophan, we must have arginine, leucine, isoleucine, valine, proline, lysine. So let's see here. Then we've got arginine. Running out of space here, so I'm going to go backwards up here. So I'm right here. I'm at this arginine. Now we're going to go to leucine, isoleucine, valine, proline, lysine. So we're going to scratch all those off. So let's see here. So we had the arginine, the leucine, the isoleucine, the valine, the proline. Now we pick it up with this lysine. So we have lysine, tyrosine, so let's write that in. So lysine, so that's, yeah, that's this one here. So we have proline, lysine. Yeah. Then we have tyrosine. That's this one. Then we have alanine. Then we have valine. Then we have phenylalanine. And then we have methionine. Just like that. And there's the tyrosine, alanine, valine taken up over there. And the last two are these ones, the phenylalanine, methionine, and that's the whole thing. If you tally them all up, you have, starting here, 1, 2, 3, 4, 5, 6, 7. Are you kidding me? Let me write that down again.
This might be a dumb question, but just out of curiosity, have you ever tried Control-Z on it?
I could try it. I'm not sure how you would do it from where I am, since there's no keyboard in here. No, there's no way to do it. The reason why it does that, I'm not the first person that this happens to. Can anybody guess why it happens to me? What's unique about me? It's not unique to only me. Nobody knows? Exactly. Somebody just typed it in the chat. That's why it happens to me. It's very common. I just got out of a meeting with a bunch of colleagues, and I was writing on my iPad in the meeting, and one of my colleagues said, "Oh, I know what happened because the same thing happens to me all the time." So that's why it happens. So tryptophan, arginine, leucine, isoleucine, leucine, isoleucine, valine, proline. Here we go. So now, if we tally them all up, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. So we have exactly all 22 residues, and we piece them together just by going backwards from the trypsin and chymotrypsin fragments. Again, we identified the valine as the C terminus. So this is our C terminus. And then the methionine would be our N terminus in this case. All right, despite my technical difficulties, give me a thumbs up if you think you could do that. Not delete everything in the middle of the problem. That's a talent unto its own. All right.
Just really quick before you move on, I'm
Yeah.
sorry that you asked. So,
Ask me anything.
When you did like the order at which you did it, I was trying to write everything down, and I did it in the correct or like the incorrect way. I kept wanting to write the whole word, so I was missing how you like the way in which you were writing everything down, or like the like where you start and where you end, and then all of your middle pieces. So, like, do you mind just like using your pointer and like going through so I can label like which one you started at?
Yeah, so the C terminus is where you start on the right-hand side.
Yep, I got that.
It started here. So we'll put here start, put down here finish. Okay, and so I went in this direction. I went down here like that, and then I came back here, and then I went down here, and then I came back here, up like that. So I had to snake it backwards because I didn't have enough space to fit it on the slide. Does that make sense?
I meant more so from the actual question, like how did you get each thing in order from what we were given in the table? I'm sorry.
Okay, yeah, no problem at all. So if we go back to trypsin, so trypsin does a cleavage of a peptide bond on the C terminus side of arginine and lysine. So we have arginine and lysine. So if you look at all the fragments, so what they did is they threw this thing into a flask and then they mixed trypsin in with it. And these are the 4 fragments that they got out. If you look at what they have in common is that 3 of them have the last amino acid as being either an arginine or a glycine. That tells you that this must have been the C terminus of all of this. Okay, it's the only possibility because it cleaves on the carbonyl side of it, and we know that all amino acids go N-C-carbonyl. So if you have an arginine, right, it cleaves on the carbonyl side of it. So that's why it cleaves on this side of the lysine, this side of the arginine. Right? It doesn't cleave in between the cysteine and the arginine. It cleaves on the end of the arginine, the end of the lysine, right? Does that make sense?
Yeah.
Okay, so,
yeah. Sorry.
okay, so everything that's cleaved by trypsin has to end in either arginine or lysine. But there's only one thing that doesn't, and it ends with valine. So that means that that's the end of the molecule. That is the C terminus. So that's why I started with this right here. If you look at the chymotrypsin fragments, it's the exact same rationale. Chymotrypsin cleaves the C terminus side of phenylalanine, tryptophan, and tyrosine. If we look here, we have a tryptophan, a tyrosine, a phenylalanine, a phenylalanine, a phenylalanine. Those are all things that chymotrypsin cleaves. The only thing that it doesn't is a valine. So again, you get the same answer from the chymotrypsin, that it must be the valine that's the C terminus. After that, you just look at the fragments. What's the C terminus attached to? If you look over here, it says that the C terminus is attached to an alanine. If you go over here, it's the same thing. It's attached to an alanine. So you scratch that off and you say, there you go. That means going backwards, the next amino acid must be an alanine. What's the alanine attached to? Well, it's attached to a phenylalanine and a proline. We go over to the next place. Sorry, a phenylalanine, proline, then a glycine. So I wrote phenylalanine, proline, and glycine. And the only place over here that you find phenylalanine, proline, and glycine is right here, where I'm crossing it with my green pen. So After the glycine here, you don't know what comes next when you look at the trypsin fragments. But when you go to the chymotrypsin fragments, you see that it must be arginine. So you go arginine, cross that off. Cysteine, methionine, cysteine, methionine. So those are gone. You come over here, you look for cysteine and methionine. Is there methionine in here anywhere? Right here, look, we have arginine, cysteine, and methionine. So we cross that off because it's already in there. So what comes before the methionine must be a phenylalanine. So phenylalanine, histidine, tryptophan. So we cross that off. We put phenylalanine, histidine, tryptophan. We go over here. We see that the only place that you have a methionine and a phenylalanine attached together anywhere is this. So we can just scratch that off. Then before the methionine, you know, what comes before that? Well, we already had all the way up to the tryptophan. So then we look for a tryptophan over here. We find the only tryptophan in the entire thing over here is this one right here. Right? It's this tryptophan. So then we cross that off, and then we go to arginine, leucine, isoleucine, valine, proline, and lysine. So we get all of these ones, right? And then we come over here, and we find the only lysine is this one here. So we go lysine, and I already lost these ones. So we have lysine, tyrosine, alanine, valine. Just go backwards like that. Does that help?
Yes, thank you so much. I was very confused. Yeah.
Yeah, it's just solving a puzzle is all it is. It doesn't really have all that much to do with organic structures. It's more like learning how to play a new version of Sudoku or something, you know? It's just looking at the fragments and putting them together. That's really all it is. Yeah. All right. Well, why don't we take a break? I think we deserve one.